We see the value of AI in doing slide QC, right? It's really important to make sure that we get rid of out of focus areas and all those typical artifacts we see in slide scanning systems. Using AI to exclude areas that are out of focus or folds. Understanding the complex tissue microenvironment. You have tumor, stroma, muscle, fat. You have all these different tissue architectures, and typically you're only interested in, at least in oncology, of measuring the expression in the tumor cells. So being able to develop an AI model that differentiates all those different tissue classifiers, or different classes, is extremely valuable. And then you move on to the cell analysis, where we're trying to detect nuclei and measure cytoplasmic expression, membrane expression. I feel like have really been a game changer, in the last 5 or 10 years of being able to accurately segment cells.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Welcome, my digital pathology trailblazers. Today, I'm here with Doug Bowman from Indica Labs, and our podcast is about companion diagnostic in the new AI era, AI-powered companion diagnostics. Welcome to the show, Doug. How are you today?
Alex. Yeah, doing great. I'm excited to be here.
So we always start with the guest. Tell the trailblazers about you.
Yeah, so my background my degree is in biomedical and electrical engineering. And, I like to start with my first job. My first job was at a medical research facility or medical research group, university hospital. And, looking back at my first job, it really was the foundation of my career in microscopy and digital image analysis. So I both learned.
What did you do there?
We had a lab that was pretty, I would say, pretty high tech, in that the principal investigator was well funded. We built all the own imaging systems that we use, so digital imaging microscopes. This was kind of in the early days of digital imaging. So we built all these high-speed time-lapse systems and 3D microscopes.
Oh, wow.
And so the group included physicists, computer scientists, engineers, optical people, microscopy people, and then, of course, the scientists that were in the lab doing their PhD research.
That's a good background for digital pathology work, because this is also very multidisciplinary.
Definitely.
Then you learn. I think this is a skill that is underappreciated in the field, because you have a lot of very skilled scientists that still need to learn to talk to each other. So that setup that you had is very much setting you up for success.
And honestly, I did not realize this coming out of university, right? I'm an engineer doing, programming and designing electronic circuits and stuff like that. And what really hit me during that first position was the research, the work. So I was basically building things to respond and help the scientists answer their biological questions, right? So my whole career has been around really using the technology to support, biotech, pharma, and medical research. Yeah.
And your role at Indica Labs?
Yeah. So I'll after that, I did spend some time at a software company before Indica. And then after that, I actually went into a large pharmaceutical company. And so that was here in the Boston area. I spent a lot of time supporting kind of the whole drug discovery pipeline. So early discovery research, time-lapse microscopy, stuff like that. And then moving more towards the translational and clinical space. So this was a little bit in the early days of digital pathology. So Aperio was kind of just starting with their slides, commercial slide scanners. So a lot of the early effort was building slide scanning systems out of microscopes. But then as Aperio came along, we built basically the digital imaging laboratory that went all the way from a limb system and sample management through scanning and histology and delivering pharmacodynamic assays to our scientists that we were working with. And then transition. So that's where I got to know Indica Labs. They were obviously a software provider, Halo. We started using that at the pharma company. And I really enjoyed working with the team at Indica Labs. And so ultimately, I convinced them to hire me and build a pharma services group.
Oh, my goodness. So you were the OG of the pharma services at Indica.
Correct. So over the past 9 years, supporting our biotech and pharma clients, we saw a real need, especially in the world of companion diagnostics, a real need to combined groups at Indica. So we now have a precision medicine group that consists of a traditional pharma services team that's supporting pharma in the services role, and then our AI diagnostics team that is building AI commercial solutions. So kind of a combine of that really fits well into the CDX story.
So you're the boss of which group?
Both. We've combined our pharma services and AI diagnostics into a precision medicine group. Yeah.
Amazing. So speaking of precision medicine, let's talk about the companion diagnostics. Let's start with explaining what it is. What is a companion diagnostic and why they are becoming critical in precision medicine?
Yeah.
And especially for complex therapies. And by complex therapies, I mean antibody-drug conjugates.
Yeah, so companion diagnostics, the audience is probably pretty familiar with it, but it's essentially what the word says, right? It's a companion to the therapeutic. So in precision medicine, as you get more and more specific and targeted therapies, the need for a companion diagnostic is really important because you're gonna help the clinician understand what patient population might respond to the treatment better. So that companion diagnostic really gets developed in parallel with the therapeutic.
So you have the test, and then depending on the result of the test, the patient either will get the therapy or not. And what are we assessing in this test?
I mean, it really depends on the therapeutic. I mean, a good example is HER2 for breast cancer. HER2, the targeted therapies for HER2 rely on having HER2 expression. So a test for that would measure HER2 expression in the tumor cells.
So we have a biomarker, and then we decide, okay, a certain level of biomarker qualifies people to get the drug, and below that level, there is no benefit of the therapy.
Correct. And that level is really important, of course. Like which level is the patient going to respond if it's low expression versus medium versus high expression.
And that's probably involved in the development both of the drug, clinical trial, and like all these combinations, right?
Correct.
The whole process of developing pharmaceutical with the test.
Right. I mean, I think in early preclinical work, they can get an idea, at least in animal models, like what the cutoff is for correlating with response and efficacy. But then, and they can model that and maybe estimate what that is in the human. But really, you need to do those tests in the human samples to really understand what that cutoff is. And that's certainly one of the challenges, I think, for companion diagnostics in general. So, companion diagnostics like the HER2 example, that is one that's traditionally read by a pathologist. Pathologists are very good at, looking at different tissue morphologies and then measuring expression. But there are challenges with some of these biomarkers, and HER2 is a good example where, more recently they've seen clinical responses with HER2 low and HER2 ultra low. And when you have really low expression, the importance of the histology and the reagent part of the assay is extremely important. But it is an area where quantitative image analysis can really excel at capturing that low expression.
And I personally have a strong opinion on like visual scorings that until we had good image analysis, was as good as we could get. But the strong opinion is basically the biology, the physiology of human vision. You can only estimate, and if you need like a precise cutoff and decide whether a biomarker level is below or above this cutoff, there's always going to be 50-50 chance that you're going to be wrong around the cutoff if you're doing it visually, right? If it's obvious, then it's going to be fast and reliable. But around the cutoff, which is where it matters, because this is where the decision is being made, is the patient going to get the drug or not? Human vision is not the most precise instrument to do that.
There's that classic example where there's a checkerboard, a grayscale checkerboard, right? And you have two like little cylinders in different parts of that. And, visually, 90% of the people are going to say those are different intensities, and then they slide the two cylinders next to each other, and indeed they are the same intensity. So the context really matters. And when you think about like the checkerboard relative to tissue, you can get really complex, heterogeneous expression in a tissue. That's a really challenging problem for a pathologist, visually scoring.
There's an interesting paper as well by a colleague of mine, Famke Effner, where she explains the paradox of ground truth paradox, especially in this area of visual illusions, of color perception. And this is where computers are a lot better in doing this. So that brings me to the next question, which is AI capabilities. So we have the companion diagnostic. It is a test to check, okay, what's the level of the biomarker, because we have a cutoff, and we are not that great in evaluating around the cutoff. So, and that was the traditional way of doing this. Also, traditionally, these tests were relying on a single biomarker, right? We are giving this example of HER2, that's just one biomarker. And now in the era of precision medicine, there's going to be more. There's going to be maybe immunofluorescence at some point. Basically, the visual image to assess is getting more and more complex. So how is AI expanding the capability of companion diagnostics?
Yeah, I think AI is really impacting all aspects of quantitative analysis. We see the need for, or the value of AI in doing slide QC, right? It's really important to make sure that we get rid of out of focus areas and all those typical artifacts we see in slide scanning systems. And they're not really inherent to the slide scanning systems, but you do get some artifacts like that. So using AI to exclude areas that are out of focus or folds, understanding the complex tissue microenvironment. You have tumor, stroma, muscle, fat, you have all these different tissue architectures. And typically you're only interested in, at least in oncology, of measuring the expression in the tumor cells. So being able to develop an AI model that differentiates all those different tissue classifiers, or different classes, is extremely valuable. And then you move on to the cell analysis, where we're trying to detect nuclei and measure cytoplasmic expression, membrane expression. The AI models, I feel like have really been a game changer, in the last 5 or 10 years of being able to accurately segment cells. If you look at a tissue like colorectal, where you've got these really densely packed nuclei and they're elongated and they're all right next to each other, the AI models are very good at capturing those. They still have their challenges. Throughout that whole workflow I described, there's challenges at each step.
I think immune oncology is like a classical example where there's too many variables. And we try to compact or you try to find like the one thing that the pathologist can assess visually, or at least historically. This was the tendency, because then you can easily deploy it in the clinic.
Right.
But for immune oncology, we know, okay, different immune cell populations, the locations of them, how far they are from the tumor margin, all that stuff. By the second variable, human vision is not good enough anymore.
Right.
So with AI, and by AI here, our application is going to be AI for image analysis in the broadest sense of this broadest sense of the meaning of the word. Basically, like AI for working with these images. Any benefits for the patients? How would you describe why is AI more beneficial for the patients?
Good question. I think ultimately it comes down to the accuracy of the test, whatever test it is, but certainly in image analysis, the accuracy of that test. You need to make sure that it's detecting, only tumor cells or as much tumor as it can. The quantitation of the biomarker itself, whether it's a nuclear or cytoplasmic or membrane, making sure that's as accurate as possible, so that when it gets to that stage where you're defining a cutoff to enroll or not enroll a patient, that's as accurate as possible. Yeah,
and that basically is a function of the lack of precision of human vision. Basically, we can overcome that hurdle with computers.
Right. Now, that's not to say there are numerous challenges in it.
Of course.
I think at this point, it's still a pathology assist tool, right? It's going to be a tool the pathologist uses to make the measurements. The pathologist is going to, see markups in images that give them an idea of what's been scored and how it's been scored, so they can, verify that they agree with the decision of the AI model.
I think it's a perfect combination of this tandem work because you leverage the advantage of the computer of calculating the exact threshold of as exact as we can get, and it's a lot more exact than with human vision. But then confirming, is it in the right place? Is it actually doing visually the job that the pathologist would do, but with better calculation? So like you said, assist tool, tandem work with the pathologist, with pathologists in the loop.
Yep.
relevant for the quality of the test itself and also for patient trust, for patient safety, I think.
Right. And I think it's important to note that the pathologist is involved in much of the earlier stages as well, right? The pathologist is involved with training the model, assessing the model, iteratively improving the model. So they're involved throughout the process.
So specifically for antibody-drug conjugates, what are the challenges there? Like, why is it more difficult to develop a companion diagnostic for this type of therapeutic?
Yeah, I think ADCs, so antibody-drug conjugate, this is a system where you identify a target population of cells. Hopefully there's some protein that's being expressed only in tumor cells.
Right.
And then the ADC has a drug toxin that's attached to it. And so it's very precise in identifying the cell population that it's attacking. So as far as companion diagnostic, it's really an ideal companion diagnostic because you know which the target population is. You can develop an assay all the way from the antibody, the reagents, scanning, image analysis workflows, specifically to measure that. In most antibodies are looking at a single target, but there are some new bispecific ADCs. So they're looking at two different targets. So there's always the question, do we develop a single assay that looks at two markers, or do we just do two in parallel, two side by side? I think we're still very much in the early stages of that. But I think time will tell as we get more and more data and maybe more and more information, if that leads to a more accurate test, then we might move to a multiplex environment or a multiplex IF environment.
Because I think we're still optimizing for, how can it be deployed without a computer, even if we have the capability. But the moment we actually have proof that this biology cannot be assessed in a different way, that's going to be the moment where it's not going to be an option and more to not do it with a computer, not do it with AI.
Yep.
So,
and I think the AstraZeneca TROP2 story, if you're familiar with that, is a really good example where they identified a relatively unique, signature that captures or correlates with efficacy. So it's an ADC, so theoretically it should be membrane bound. So you would think that just membrane expression would really drive that scoring criteria. But what AstraZeneca found was that it was a ratio of the cytoplasmic expression and membrane expression. So that's a case where image analysis fits right in. It can analyze those different subcellular compartments, create that ratio that you can then create the score.
I am very excited about this one because this is exactly and even without going multiplex, going multiple markers, it's the same marker where there is this visual thing that a pathologist cannot calculate.
Very challenging for a pathologist to calculate a ratio in their head while looking at all the heterogeneity in an image. Yeah,
I know. So this is a perfect example of that.
And it falls really well into image analysis because, image analysis is generating a lot of different metrics with each image. It's not just expression. It could be things like nuclear shape, nuclear size, cell morphology, cell size. So there are a lot of features that get extracted out of an image analysis algorithm. And you never know, one of those might be important for, correlating with outcome.
And it's a little bit there's never a guarantee that you're going to find this biology in there. But if you don't look, then you're not going to find it. And here they did look enough to find something that was better than just a visual cutoff.
Right. Yep. And I think that actually introduces one of the challenges with development, right, is they don't know if there's going to be a predictive score. But in order to try to drive that part of the CDX development earlier, they want to start that at risk earlier in that development. They can't wait to the end of the clinical trial then to go back and do the CDX. They need to do those in parallel.
That is true. And I always when I remember this test, and then I think, how many of other biomarkers were tested and how many drugs maybe have failed the clinical trial because there was not enough granularity there wasn't a method to extract enough granularity out of the data to actually find some signature. And it's, it's always a funnel. You're gonna, I say life is a funnel because there is always so much trial and error that you have to do at the top of the funnel to actually get to something. And there's never a guarantee that you're going to get to something. But there is a chance. So definitely leveraging AI to find something that can later help patients justifies this investment, in infrastructure. And you mentioned that when you're going to be working on an AI-based companion diagnostic, it's going to be a system. You said scanner, and the lab work for IHC, and it becomes an end-to-end workflow,
Definitely.
right?
Definitely.
It's not just like IHC and you show it to a pathologist.
And I agree that biomarker-driven clinical trials are going to become more and more prevalent, whether it's just to confirm, mechanism of action of the drug itself, or to look at outcome, or, what patient population for patient stratification. I think it's going to continue to evolve and grow.
I think with this, I don't know, proof of concept, you can call it proof of concept, that it actually is there, because until this very thing that they found out, I was like, oh, maybe it's there, maybe it's not there. Everybody's looking for the holy grail of signature, and they actually found it. So it's worth trying. But it's a workflow, it's a system, and that brings me to the partnership that Indica Labs have with Leica.
Yep.
Can you tell us a little bit about that?
Yeah, I'd love to. So back in January of last year, 2025, we did a strategic partnership with Leica Biosystems, and it was really twofold. It was around our clinical product, Halo AP. So being able to deploy these algorithms in the clinic or the diagnostic labs. So that's Aperio Halo AP DX is the software platform there. So really focused on the clinical market, and then combined with our AI diagnostic and AI clinical algorithms, being able to deploy that in the clinical market. But secondarily, and really interesting to me, is the companion diagnostic, right? Leica is a leader in that end-to-end solution where Indica fits in really nicely. They can do all the reagent development, they do all the immunohistochemistry and protocol optimization. They have slide scanners. They have the GT450DX, which is an FDA-cleared slide scanner. They have a global footprint. So combining that capability with Indica Labs and AI development really allows us to offer that end-to-end solution. And I think it's especially true in companion diagnostics, where there's going to be a quantitative component.
Very much. And you have a history with them, right? Because the founders were previously at Leica, and then they span off to work on Indica Labs, and now there is the collaboration back to work together.
Yeah, correct. Our founder, was it back in 2011, left Aperio and built Indica Labs. Yeah, it's quite exciting, actually. I think, and you'll see that at the company in general. A lot of us were in the area, whether they were at biotech or pharma or in academia, using the products, using image analysis, and then have joined the company afterwards.
It's also a good fit. Because of this heritage, you kind of understand their technology, and like you knew what they were doing. It's not like trying to fit to each other without knowing what it was before.
Right.
So
there are a lot of image analysis is really prevalent, but I think the domain knowledge, and when you get a certain tissue type or challenging images, really understanding what those challenges are and what the best solution or what path forward is to try to optimize and remove like artifacts and stuff, that experience is really powerful.
So when the decision is made to develop an AI-powered diagnostic, how does that actually work? What does the journey look like from early development to regulatory approval? And I want to cover it for like now for single biomarker, but also how would it look in the future if we had multi-model, multiplex, different data modalities flowing into this, because that's an option as well,
it's
right?
Definitely an option. And I think that's the power that we're going to see with precision medicine in general, is being able to bring in different assays that add to that story. So the development journey, it's interesting because at least for quantitative image analysis, companion diagnostics, right, there's not one on the market yet. So we're still very much early in that journey. But the workflows and the kind of progression of assays from early development, early discovery through development, stuff like that has been going on for a while. It's just that last piece that we're kind of waiting on. So we typically see these image analysis algorithms being developed in the preclinical models. So many of our pharma customers have our software. They're developing the algorithms maybe internally to help drive their preclinical work, maybe their lead optimization, for example, when they're looking at different preclinical models. So they're developing the algorithms or some version of the algorithms earlier. As they start moving into the clinic, that's an opportunity where they might come to someone like us to further develop that algorithm, maybe do a feasibility in human samples, modify or develop a new algorithm and train that algorithm, for those clinical samples. And that early feasibility allows them to get like an early story, like, is this going to work in the human setting? And then as they start seeing promise there, then you would want to start further developing the algorithm more towards a research use only or clinical trial assay, and seeing if they start seeing results in the clinic. Okay. Both therapeutic, right? This is going along with the therapeutic. So the biomarker assay might work great and the therapeutic does not. So all of these things are working in parallel. But as that therapeutic progresses, and especially in the idea around companion diagnostics, and ADC is a good story for that, at some point at risk, you need to start developing that ADC companion diagnostic in parallel with the therapeutic. So we would be further generalizing the assay, looking at different scanners, different sites, different analytical validation, clinical validation, kind of in parallel with the therapeutic development.
And then once you know it works, then it's going to be the regulatory path of that.
Yeah, exactly. I mean, we want to start that regulatory process as early as possible. For AI development, that's typically around the documentation and quality management system that you have while you're doing the development. So we would typically start the design history file, doing all that documentation a little bit at risk earlier, so that if it does lead more towards the regulatory process, we've got a jump start on that, and it's going to be a lot quicker to get there.
And the documentation is a keyword I hear from regulatory consultants and everybody who's helping companies do these clinical trials or for devices. That is document from the get-go, because you cannot go back to redocument.
It's very challenging to go back, right? I mean, you need to document where your training images came from, the different versions, the iterations that you might be doing throughout development, making sure you have the right, target demographics in your training data for what the clinical trial is going to be. And the sooner you do that and the more data you collect, of course, the stronger and the better the model you'll have. But ultimately, you'll be farther along when you move towards that regulatory process.
Definitely. And what about the multimodal? Does the development look different?
I think the development maybe is similar, but it just adds complexity if you've got more than one measurement. An example I like to use is in the ADC space, there's something called the bystander effect. So the bystander effect, when the ADC drug hits the cell, gets internalized, the toxin gets released and kills that tumor cell, you end up with free toxin. So that cell dies and you get free toxin. So if you have a neighboring tumor cell next to it, that toxin might end up killing that and you'd get a bystander kill, which would be a positive effect, right? So being able to quantify that can add some value to that scoring metric. And we do that through what we call spatial analysis. So you're measuring the expression level of those neighboring cells and its proximity to the target positive cell to get a score about if there's more tumor cells close to those tumor target positive cells, then you're more likely to have that bystander effect, and you're going to be more efficacious for that therapeutic. But it is a different metric. It's a different scoring metric. It is one that's very challenging for a pathologist to read, right? Trying to capture the spatial relationship of all the tumor cells. So it bodes well for image analysis to bring that modality in as well. And ultimately, it's just going to be part of that scoring metric, right? That scoring, that predictive score is going to be a combination of membrane expression, cytoplasmic expression, and maybe the proximity as well.
I remember the story of Immunoscore, where they were measuring was it CD8 and CD3,
the cytotoxic T cells that were close to that tumor margin.
Exactly.
Right.
So that's already something you cannot do visually, and it ended up being a image analysis driven score for colorectal cancer prognosis. So.
Yeah, I mean, that was really exciting, I think, for the image analysis world too, right? Something that you can't capture, visually, or very hard to capture visually. And from an image analysis perspective, it's a relatively simple calculation, right? You're looking at all the immune cells, you define the tumor border, and then you just measure that infiltration region. One of the challenges is the fact that you're going through the regulatory process with the therapeutic, with the AI algorithm, with the actual cutoff that you're working with, right? Trying to understand what that cutoff is. All of those are going through that regulatory process in parallel.
Any other considerations for regulatory? What else is challenging?
I think with ADCs and companion diagnostics, the biggest challenge is that the the diagnostic, right, the biomarker assay needs to go through that regulatory process in parallel with the therapeutic. So, the therapeutic, you're looking at adverse side effects and efficacy. And meanwhile, you're going through the biomarker process where you're developing the assay, you're validating the assay, both analytical and clinical validation, and then going through that regulatory process in parallel and somewhat at risk, right? Yes. So it's just a lot more documentation needs, a lot more communications back and forth between, your diagnostic partner, the pharma company, and the regulatory agency. So, that relationship is obviously extremely critical for any biomarker development, but especially companion diagnostics.
Yes, the relationship aspect, another keyword, I sometimes highlight these keywords, the relationship, because you have compounding of complexity. Right, there's already enough complexity in filing for a drug. Now you have the test. You have people from the pharma company. You have people who are maybe software developers in one way or another, image analysis specialists, the lab people. So a very complex landscape that needs to be coordinated enough to later be able to file the necessary documentation. So definitely important to
and I think finding that
pick
partner,
the people you want to work with, right?
I mean, both from a project relationship, right? You need to have trust that they're going to be able to execute on the technical aspects, but then also the regulatory aspects. And it is one area we see our partnership with Leica being one of our strengths is, we have that experience not only from a technical aspect from end to end, but from the regulatory aspect as well. And even within our AI diagnostics group, we do have experience going through that regulatory process. We work closely with our regulatory group. So we have a CE marked algorithm. We have a 510K cleared Halo AP, which I mentioned earlier. So we've already gone through that kind of regulatory process on a couple of our products. And obviously looking forward to doing that with the CDX.
Yes. And I think it's a process. It has definite steps. So anybody who wants to do that can go through the process, but the experience of having done it is invaluable.
It is.
And on both sides, both companies have that experience, right? For different things. This is a different way of thinking about your products once you're in the regulatory environment and you're trying to get something cleared, something maybe approved.
And I think ultimately, when you're earlier in the process, you're trying to decide between, a manual visual read versus, a singleplex biomarker assay, or maybe a more complex measurement. So you kind of want to go through the process in parallel with that as well, because if it's a simple on-off type biomarker, then there's probably little reason to do the quantitative analysis. But if the quantitative analysis adds value, then obviously you want to take care of that too. And then having a platform that you can deploy that, at a diagnostic lab is important as well.
Definitely. I think the availability of options doesn't mean that you need to take advantage of all the options. And what I mean is okay, if you can throw all the markers and do a multiplex, it's going to be challenging to deploy. So it's kind of a balance. The least complicated, the least complex that gives you the most benefit. And sometimes it's going to be a simple marker for visual assessment, and sometimes it's going to be spatial relationships between different immune cell populations, right? So.
Yeah, I think that's one of the biggest challenges, just what that added value is, and making sure you can come up with that decision and story.
And I think that makes image analysis now, it takes it from the just specific application level into the level of this is an acknowledged method, scientific method that should be applied. And then the scientists define, okay, what's going to be the application. But image analysis is something that needs to be leveraged because you're not going to find it out any other way.
Right.
Which makes me excited.
It's very exciting.
I come from the image analysis background as well.
I mean, for me, obviously, being able to impact patient care is the ultimate goal of everyone that's working in this field. But personally, my kind of career path, where I kind of started off in basic research, and then now I'm going to have, potentially have an assay that is impacting a therapeutic. That's super exciting.
I know.
Yeah. Especially with digital image analysis and microscopy, with all the technologies that I've grown up with.
I know, it's full circle
Yep.
moment.
Definitely.
You know what else is important? That ties back to the user group meeting that you have today, is the image analysis literacy. Because now that it is becoming this established method, it used to be like cutting edge, who liked, the scientists that liked it would get involved in it. But now it's similar to molecular pathology.
Yep.
When you become a pathologist, there's no option to not learn molecular. This is part of the patient care.
It's a fundamental tool that everyone needs to be using. Yes.
And I see image analysis like that right now as well. And when we were coming to this podcast, when we were walking, and we were talking about, when I was doing my image analysis work at the image analysis company that I was working at, the crucial thing that we had to start with was the communication between pathologists and image analysis scientists, the different image analysis concepts that the pathologists needed to learn.
Right.
So like the segmentation, classification, and all these computer vision, high level computer vision terms, just to be able to communicate with your scientists and basically do a joint work of creating image analysis solution that actually matches the tissue.
Yeah, and I think that's where we call them application scientists, but they're the application level people. These are typically people that are really experts in image analysis, or experts, they've been in science labs and biology degrees, but they're that kind of middle person, right? They can understand what is needed in the industry, what algorithms are needed, what tools are needed, and they can convey that back to our development team so they can develop those tools. And that's one of the really values of our user group meeting, is we get the users together. We hear, of course, we're presenting to them some of the new features of our software and giving them some information about our software, but we're also having some users present to see what they're doing. Other users in the audience can understand how other people are addressing certain challenges. And then, of course, all of those ideas are feeding back into our development team so that we can improve upon our product, or continually improve our product.
And I looked at the names at the registration table. There was people from pharmaceutical industry, academia, some private research places. I don't know, basically a big breadth of, big variety of image analysis application spaces.
Boston, Cambridge is like a hotspot for that. You've got dozens and hundreds of biotech companies, large pharma. You have CROs that are actually running the facility, or running the laboratories. So it's a really kind of a hot pot of technology and excitement.
Nice to host the user group meeting.
Right. Yep.
So what do you think is going to be the future? What's relevant for the future for people who are starting in this field? Because now that this is an established method, that we have proof of concepts of image analysis being used in the companion diagnostic space, there are still more people that have not done it than those who have done it. There's going to be a lot of beginners.
Right.
And how do we deal with that?
Yeah, I think the education of the next class of scientists is always extremely important. We see that partly because we have a lot of our software in academic labs. So a lot of these research labs are using our software to answer a lot of these questions in the early research. Our user group meetings are kind of like an educational opportunity as well, because if you're not even a user, it's not just a customer, it might be a prospective customer. They can come and listen to the talks too. So I think it's extremely important. I think it's more and more common. People in graduate school, even undergraduate school, are getting exposure to image analysis algorithms earlier and earlier. I think, there's a lot of other organizations. There's your podcast, right? An excellent resource for people learning about the different technologies, the buzzwords, just understanding what the buzzwords mean. There's groups like the Digital Pathology Association that puts out white papers and educational papers. I think all of that just continues to grow.
Anything specific that you're hopeful for in the future?
Well, I think first would be a digital CDX. That would be super exciting.
Yes, I'm waiting for that as well.
I think there is so much value in the multiplex technologies, whether it's multiplex chromogenic or multiplex fluorescence. I mean, in a simple case, if you have a bispecific ADC, you've got two targets. So you need to have two assays that do that. In the immuno-oncology space, just trying to understand the immune microenvironment and the relationship between all these different immune populations relative to the tumor microenvironment. That work is going to continue to expand. And I think as soon as they start identifying these multi-parametric signatures on these multiplex panels, it's inevitable that it's going to move into the clinic. As you mentioned, there are a number of challenges pre-analytical variables, the wet lab work, a lot of challenges to overcome, but the technology's there.
Mm-hmm.
Yeah.
Yeah, and the challenges, maybe they will disappear, but for the nearest future, probably not. So we have to figure out how to overcome them to get to the insights from the biology that are behind.
I think from an AI model perspective, there's new AI models continually coming out. As a company, we're assessing different models. There's foundation models where you've developed with a lot of training data, you've developed like a core model that works for pathology. What we've seen is that is a nice starting point to customize that training. So if you're going into a specific indication, instead of starting from scratch, we can start with some kind of base pre-trained model and then add training to that. So I think the models are going to continue to evolve. The training sets, getting access to training sets is one of the big limitations. We're going to continue to build relationships with pharma companies because a lot of this data is sitting in a pharma space. So collaborating with them to continue to improve these models.
Thank you so much. Thank you so much for the insights into AI-powered companion diagnostics. And if somebody is interested in Indica Labs, exploring what you guys are doing, where can they find you?
Look on the web, indicalab.com. Obviously, the website has a contact form. We have info@indicalab.com. And obviously, reach out to any of us at any time. We have application scientists spread across the globe. We'd be glad to answer any questions.
Thank you so much, Doug.
Oh, thank you.
And I hope you have a great day, great user group meeting. I'm going to be there as well. And I talk to you in the next episode.